So it's my pleasure now to talk to you guys about the gene therapy that we have for FOXG1 and the other programs that we have in development. So I'd love to have Guy come up to the stage and Sue, and let me just quickly introduce them. So Sue, can you, well, come on up, you don't have to raise your hand. If you guys haven't spoken to Sue, please do. Sue is mom to Yuna, a beautiful child. How old is Yuna now? 14 years old. We just saw her when we went to Buffalo. You know, Sue's story is pretty incredible because she is a neuroscientist, as is her husband. They work together, which I don't know how any married couple does that, but they make it work. You know, Sue was studying the Fox family of genes. And so when Yuna started having all the symptoms that she had, and Sue can go into it, she had an inkling. And then she got the diagnosis of FOXG1. And unlike the rest of us, she knew exactly what that meant. And it is coincidence, irony, I don't know what it is that this happened to Sue, but thanks to that, she's been able to change our life. And as Terry mentioned, you know, he was able to get Steve Gray, who was on our advisory board, to really focus. We were really lucky to have Sue dedicate her life to this cause. Guy Aolon, come up, Guy. So Guy has had a 20-year career in biotech and biopharma. He's worked at companies like Genentech. He was recently at Ultragenyx, where he worked on the clinical trial for Angelman syndrome that I talked to you about. And I met Guy in a totally different context. We were working, and Terry might remember this, he was helping us with this. We were trying to put together a proposal for ARPA-H to really focus and put $1 billion, you know, at least $1 billion towards rare disease drug development. And we spent so much time on this amazing effort, and of course they rejected it. So, but the highlight of that is that I got to get to know Guy, and he became really passionate about what we're doing, and he decided to leave his job where he was making a lot of money to take a job where he's making not a lot of money, and really take this program. And Guy truly believes that right now what's happening is that many biotech and pharmaceutical companies drop rare disease programs, and Terry can attest to that. And so the best way for us to make sure that we can get this drug to our kids in the shortest time possible is that we as a foundation take this forward to phase 1/2 clinical trials. And that is why we're trying to raise this money and do it ourselves versus licensing it out to a biotech right now. And we're lucky to have Guy leading that cause. So with that, I will let you guys take it over. Sue, do you have a mic? Do you want a mic? Do you have another one for her? I can give her mine. Okay, great. Okay, perfect.
Hello, everybody. It's so exciting for me to be here to share our recent progress. My name is Soo-Kyung Lee. I told you 2 years ago to stay hopeful. Something is coming, and then I'm so happy to report to you what progress we has made and remaining hopeful what change it made. My name is Soo-Kyung Lee. I am Chief Scientific Officer of FOXG1 Research Foundation, and I'm also Director of FOXG1 Research Center at the University of Buffalo. So FOXG1 syndrome is essentially caused by mutation in one single allele of the FOXG1 gene, which arise mostly spontaneously. So our mission in FOXG1 Research Foundation, as stated before, is to find a successful cure for the FOXG1 syndrome and related neurological disorder. We have this big mission and we broke down this mission into 3 parts when we are starting the foundation research program. Number 1 is making the model system. You heard from Terry that it's important to have model system that accurately mimic the FOXG1 syndrome condition for genetic condition, as well as the mice that show the FOXG1 syndrome-like symptoms. Using this model, we wanted to understand the disease mechanism, why this mutation lead to disease in our children, and ultimately we wanted to develop the FOXG1 syndrome therapy. So we made a mouse model, that has the genetic condition of FOXG1 syndrome. We introduced the mutation into the mouse of exact mutation that one of our child or many actually children in this community have. And in addition to one particular model, we also generated multiple mouse models that we represent distinct genetic mutations as well as sometimes different type of symptoms. And why it's important? It is important because we cannot open up the FOXG1 brain. We have some ideas from the MRI result that our children have smaller head, and sometimes there's a larger ventricle, there's myelination problems, but we really don't know exactly how that happens in the brain. So the mouse give us incredible opportunity that we can section their mouse and see what's going on. But you would say that, oh, those are mice, they are not humans. How about the human cells? So to complement the mouse models, we also generated human neurons that are directly coming from patient cells. So we took the cells from FOXG1 patient, and then we generated a stem cells that will, that's a very powerful cells that can be any cells in our body. And we made these stem cells as neurons so that these neurons are like neurons from FOXG1 patient cells. And this is exact complementary system to mouse models. So we have armed with now the animal models as well as human neurons, and using the advantage of these two complementary systems, we have been investigating why FOXG1 is occurring. So what happens in FOXG1 syndrome brain? Now we have opportunity to cut the brain out and then see what cells we are missing, what connections we are missing, why all the symptoms was arising from this mutation. And most importantly, this allows us to ask, how do we fix FOXG1 syndrome? Okay, thank you. So it could be this is normal child, it could be we are missing some cells, it could be cells are not insulating properly within the brain, so the neural signal is not going well, maybe the connections are not as many as the children with neurotypical brain, or there could be too many connections. So as stated by Nasha, our problem is very daunting, that we want to develop FOXG1 syndrome therapy. When we are studying, we don't know what is FOXG1 brain is like in the mouse, what the FOXG1 neurons are like in these human neurons, but the goal itself is very well defined. We have too little FOXG1 in the brain of our FOXG1 children. So the normal brain, neurotypical brain, has 2 copies of FOXG1 gene driving FOXG1 levels for 100% of FOXG1, but our FOXG1 children has only 1 copy of FOXG1 gene, so they don't have 100%. They have just half of the levels of FOXG1 of normal brain, and that's why we see all these symptoms in our children. So we need to deliver the functional FOXG1 gene into the brain, which will push the FOXG1 level to 100% in their brain cells. We are targeting specifically brain cells because that's where the problem arises. It's very clear from where FOXG1 is present in our body. It's also clear from our mouse model studies. Okay, so how do we deliver the FOXG1 gene into the brain cells? So we turn to the gene therapy strategy using the virus called AAV9. So we constructed the virus that has FOXG1 gene, functional FOXG1 gene in it, so that it can deliver human FOXG1 gene into the brain cells, which will then push FOXG1 levels into 100% in the brain. So we got this virus, we made this virus, and we know that this virus can express FOXG1. And the next test is, how do we test whether this virus is working to fix FOXG1 brains? And that's where the mouse model system play a big role. We made these mice that have FOXG1 syndrome-like symptoms. that's happening in the brain, that's happening at multiple levels of their mouse lives. So we injected the FOXG1 virus directly into the brain of the neonatal newborn mice. So it's getting injected into the brain, and then we give them a chance to express and push FOXG1 levels in the brain. And a week later, this is how long it takes in mice to get the higher level of FOXG1. A week later, we check how these mice talk. Mice don't exactly talk like humans, but they make the sound that we can quantify, and we can detect and quantify. And a month after the injection, we can take their brain out and then see what happens in their brains. So it's important to understand this model system first to know that whether these mice show the FOXG1 symptoms. So I'm comparing here human FOXG1 syndrome symptoms against what we see in the mice. So as all of you know, that FOXG1 kids have small brain. And they have also cortex that's not developing quite like healthy children. And FOXG1 mice, just like FOXG1 patient, it has small brain, and it's missing many of neurons that should be critical to send the neural signals in the brain. FOXG1 children have smaller, underdeveloped hippocampus inside their brain. FOXG1 mouse exactly have the same problem. Their neurons are not insulated correctly in FOXG1 children's brain, and then we see that in mouse model as well. They have certainly communication and social deficit, and the mouse model also do not make as many sound or as much sound. Okay, so let's take a look at this. This is supposed to be the black background, so we are missing a lot of colors here. Let's see whether we can make some of result visible. So here, what we did is first we injected these mice with AAV9 FOXG1, and then we looked at their brains a month later, which is about adolescent stage of the mice. The mouse brain looking like this. If we cut the brain out, you can see the brain here, cortex and hippocampus. This is depicting where mouse function is expressed. So these are very green signals that shows FOXG1. Now we look at where FOXG1 human function is expressed after injection, and this red signal shows very similar expression pattern like mouse FOXG1. How about the protein levels? We look at the FOXG1 levels in the brain. This is the normal mouse brain FOXG1 level. So let's say this is 100%. FOXG1 mutant mouse has lower level of the FOXG1 as expected. Then this level goes up to quite normal levels after gene therapy vector injection. So that's great start. How about the neurons? So we looked at the cortical neurons, which is greatly reduced in FOXG1 mutant mice. So this little dot shows the FOXG1, this little dot shows the cortical neurons, and we don't have as many neurons in the FOXG1 mice. But after gene therapy, we were able to recover these neuron numbers to the level of the normal mice. This is quantified here. There are less neurons, then these neuron numbers increased after gene therapy. Let's take a look at the hippocampus. So hippocampus is the brain structure that's in the deep inside the brain, and it's a very critical component in the brain that's for learning and memory. So when you don't have correct hippocampus, there's cognitive deficit. And we look at the hippocampus structure. So we looked whether there's the correct size of angles in this hippocampus structure. In FOXG1 mutant mice, the angle is increased. That means they are not formed correctly. With the gene therapy, in these mice, exactly these mice, we were able to see that these angles are corrected, meaning that we are getting now normal looking hippocampus structure. How about insulation? So the nerve cells here should be insulated correctly, so you're not losing the neural signal when there's a firing of the neurons, and it's very critical. And then what we look at is this process of generating insulation. So here you get immature cells that are not insulating, but these immature cells eventually become the cells that can initiate these nerve. And we looked at these immature cells in the FOXG1 brain. So unlike normal brain, FOXG1 brain has increased the number of these immature cells quantified here. With the gene therapy, this goes back down to the normal brain levels. So we get abnormal higher number of immature cells in FOXG1 brain, and we can reduce it to correct levels. How about then actual insulation? Are we insulating our nervous cells after the gene therapy? That's what we are looking at here. So this is the normal brain. Each fiber represents that neurons are correctly insulated. And you can see that there's nice looking fibers going reach to the top of the cortex. In FOXG1 mutant mice, now you don't have as much insulations. So you have these little white signals, the fibers are not very good, and they're fragmentized. And in the same mouse, when we inject the gene therapy, you see that remarkable upregulation of this insulation. So we are getting this insulation back after the gene therapy. So all this together, it points out that we are making a lot of fix of FOXG1 problems in the animal's brain. How about their speaking skills? So we can take the mouse pups when they are 7 days old, and we take the mice, these pups from the mother, then the mice are calling for mom, and we can quantify that signals. So compared to the normal mice, their calls from these FOXG1 mutant mice are shorter. And then after gene therapy, it gets recovered so that their calls getting elongated. So we do see the communication skills get improved in the mice. So putting this together, we have the symptoms in FOXG1 syndrome. Our mouse models recapitulate a lot of problems. And then after gene therapy, these are all corrected inside the brain or their communication skills. So we wanted to test this idea in more our case treatment condition. So we made the two changes. The first one, we are using this W mice that has exact mutation of children, FOXG1 child. And then we also do the injection at a month-old mouse, which represent adolescent mouse. So instead of using newborn mice, we are using older mice now. And then we did inject our AAV9 vector directly into the brain. And then a month later, then in which the mice become adult, we take these adult brains and see whether the FOXG1 syndrome condition was repaired. So here, I will give you 3 examples. This is hippocampus, again, the structure that's so critical for the memory and learning process. Essentially, cognition requires the intact structure of this hippocampus. So we are measuring the hippocampal structure, this angle. Here's the normal mice, and in W mice, which mimic the patient mutation condition very precisely, this angle gets larger, and after gene therapy, this goes pushed down toward the normal mouse levels. Then how about insulation? I told you that insulation is required for normal neural firing and then for neurons communicate each other. So we looked at these immature cells. Here's the normal mouse. In W mouse, again, FOXG1 syndrome mouse, they showed increased immature cell numbers, and this is fully recovered after the gene therapy. So they don't have this immature cell, too many immature cells after the gene therapy. How about actual myelination, which is the process that initiates this nerve? And here's the result, that compared to control, the myelination is reduced in W mice. With the gene therapy, it gets recovered to the normal levels. So now we fixed a lot of things here in the brain. We get more neurons, hippocampus are looking good, the neurons are properly insulated. Then we did look at the inflammations. So this is new finding that nobody has reported before, but we found that the brain of FOXG1 syndrome are inflated. So the immune cells in the brain is activated aberrantly in FOXG1 syndrome brains, and then these active, angry immune cells can create a very neurotoxic environment that will kill the neuron cells. So that's represented here, that compared to control, we have increased immune cell numbers in the FOXG1 brain. It's not only increased number, they become angry, attacking neurons. And then with the gene therapy, we can calm down these immune cells, that the number gets reduced, immune cells are not angry, and this is the important process to recover the toxic environment in the brain by gene therapy. So again, going over this together, we have many issues in the brain of human FOXG1 syndrome. The mouse models allow us to look into the brain directly and identify individual neurons and see what happens. And then all these human problems is recapitulated in the mouse models. And after the injection of AAV9 FOXG1, we have seen all the recovery of each of these process. So this result really is a really good promise that AAV9 FOXG1 would be an effective treatment for FOXG1 syndrome. It's almost impossible for me to believe how well everything went. So here, FOXG1 Research Foundation, we set out the missions. The specific missions are we wanted to model mouse and human models that exactly mimic the FOXG1 syndrome symptoms. Then we do understand the disease mechanisms, such as the brains are very toxic environment. FOXG1 brain makes very toxic environment. And then we use the gene therapy to show that each of these disease process is blocked or repressed by FOXG1 gene therapy. So I'm very happy to report this 2 years later, after I tell you that we made the gene therapy vector, and we'll be testing whether this is working in the models that we built. So mission accomplished, but of course, just like Mission Impossible, you get more missions. So our mission continues as we advance toward the cure for the FOXG1 syndrome. So there are ongoing studies right now in our lab. We are doing dosage and toxicity tests in mouse models. It's very essential to find effective yet safe therapy conditions. And then we are also looking for the markers. It's important to understand the markers in the body of our children so that we can use the markers to assess the treatment success. Because after the gene therapy treatment, we should be able to quantify what progress they are making. We are also expanding FOXG1 syndrome therapies beyond the gene therapy, that we are looking for the small molecules or ASO therapies to see whether they have same efficacy in curing the mice, then which can be translated in humans. And we care for all FOXG1 disorders with all type of mutations. So we cover all types of FOXG1 mutation for the drug development. So this is my lab. These are very dedicated people. There are over 20 to 30 people, depending on how many work we have. Sometimes we have more people. These are incredibly motivated people. They know that they are a human face after or attached to their research project. Every single progress that they are making makes us getting close to the development of FOXG1 syndrome. This is our daughter, Yuna Lee, with FOXG1 syndrome, with my lab members, and they know that one day they can cure FOXG1 syndrome in Yuna, and we are also looking forward to the day that she can be more functional. this is my husband, Jae. So we, we make a very great team. Jae is watching over from live stream, so I had to make this comment. Okay, so we opened FOXG1 Research Center. It's a very unique situation in academia that you make a center fully dedicated a rare disease. This is our dean and vice provost, the UB, University Buffalo. Leadership is fully supported of our FOXG1 Research Center, and our goal of finding cure for the FOXG1 syndrome is supposed to say foxg1center.org, and we are sending out the newsletter to report you the progress of research that we are making, so please sign up for the newsletter. And thank you so much for FOXG1 Foundation, and thank you so much for your support. FOXG1 community has been the backbone of our research and also the backbone of our family. Thank you so much.